Hey everyone, this is Amna, your Thinker Zone buddy. Today I am back with a very informative video on glucose, glycolysis and gluconeogenesis. Do you know there's one molecule that fuels your brain, muscles and almost every process in your body. That molecule is glucose, the ultimate energy currency of life. In this video, we will explore what glucose is, how glycolysis break it down to release energy, and how gluconeogenesis build it back when energy is scarce. Stick with me till the end because by then you will understand how body literally runs on sugar. First of all, let us discuss what is glucose. It's a simple sugar having molecular formula C6H12 and O6, found in food like bread, rice, fruits and honey. Absorbed into the bloodstream, blood glucose, brain depends almost entirely on glucose. Fun fact about is, normal fasting blood glucose 70 to 100 mg per deciliter. Here is the structural formula of glucose. Now, let us discuss the sources of glucose. The basic source of glucose is carbohydrate, which includes bread, rice, pasta and fruits. These are the major sources of glucose. Next, glucose is also stored in the form of glycogen in liver and muscles, and it is released when needed. For example, in the case we are fasting, we need glucose to run our brain and even to do every single thing. So where do this glucose come? This glucose is basically coming from the glycogen which is stored in the liver and muscle by the breakdown of this glycogen. Next, we are going to discuss the glycolysis. So this is the brief introduction of glycolysis. Where do glycolysis happen? Its location is basically cytoplasm, and the whole process which takes place is the breakdown of one glucose molecule. Glucose molecule is basically a 6-carbon molecule into 2 molecules of pyruvates, which are 3 carbon molecules, and the output is 2 ATP, which is the energy, 2 NADH and 2 pyruvates, and the output is 2 ATPs, which is the energy, 2 NADH and 2 pyruvates. Next, there are the phases of glycolysis. There are basically 3 phases of glycolysis: investment phase, cleavage phase and payoff phase. In investment phase, we use 2 ATPs. In cleavage phase, 6 carbon molecule splits into 2 3-carbon molecules. And in energy payoff phase, it gains 4 ATPs and 2 NADH. It means energy is also formed. So here I added the complete cycle of glycolysis, which is glucose is converted to glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-bisphosphate, glyceraldehyde 3-phosphate, dihydroxyacetone phosphate 1,3-bisphosphoglycerate, 3-phosphoglycerate, 2-phosphoglycerate, phosphorinol pyruvate and then lactate. You can also remember this by using the mnemonic good girlfriend, fill glass daily by 3 peg and 2 peg and play ping pong and ludo. Now, let us discuss what we get from the glycolysis. The basic products of glycolysis are 2 ATPs, 2 NADH and 2 pyruvates, as I discussed before. Pathways of pyruvate are aerobic and anaerobic. By aerobic pathway, the cycle runs a Krebs cycle, and by anaerobic pathway, in which oxygen is not present, lactic acid pathway runs. For example, in case of heavy exercise where our muscles are not getting enough oxygen, muscle cramps occur. These muscle cramps are due to lactic acid accumulation into the muscles. These are some clinical relevances of glycolysis. Anaerobic glycolysis means in the cases when we are not getting enough glucose from the food or anything, or if we are not eating enough food, we will get the energy from the stored glycogen into the muscle. This glycogen is converted to lactic acid, and this lactic acid is then converted to glucose, which will provide us energy. Lactic acidosis in oxygen deficiency. In the case there is no oxygen or less oxygen in the environment, we will get energy by the lactic acidosis. Cancer cells use glycolysis at high rates. The next topic we are going to discuss is gluconeogenesis. Gluconeogenesis is basically the formation of glucose from non-carbohydrate sources. As we discussed before, glucose was coming from carbohydrate sources like bread, sugar, etc. But in the case of gluconeogenesis, it is coming from non-carbohydrate sources. The location of gluconeogenesis is liver and kidney, activated during fasting, exercise, and starvation. So there are some substrates of gluconeogenesis: amino acids for protein, lactic acid for Cori cycle, and glycerol from fats. So let us know the detail of these processes. First of all, we will discuss the amino acid cycle. The protein which is stored into the muscle is broken down by the process called proteolysis. By the process of proteolysis, it is converted into a protein such as glutamine. Glutamine then goes to the gut, where it is converted to alanine, and then alanine goes to the liver, where it is converted to glucose, and then new glucose is formed. The second phase is lactate, which is Cori cycle. It starts from the glycogen which is stored into the muscles. The glycogen stored into the muscles is broken down by the process called glycogenolysis. By the process of glycogenolysis, glycogen is converted into lactate, which goes to the liver, and then it is converted to glucose to provide us the energy. The new glucose is formed. And then we will discuss about the glycerol. Triglycerides stored into the adipose tissues are broken down by the process of lipolysis and are converted to glycerol. This glycerol then goes to the liver and then forms the glucose, where new glucose is formed. Next, we will discuss the key enzymes in gluconeogenesis, which are pyruvate carboxylase, phosphoenol pyruvate carboxykinase, fructose 1,6-bisphosphatase, and glucose 6-phosphate. Gluconeogenesis in action provide glucose for brain and RBCs, saves lives during fasting and starvation as we are not getting enough glucose from the food. Prevents hypoglycemia. Hypoglycemia means the lowering of the glucose level into the body. Now let us have a brief difference between the glycolysis and gluconeogenesis. Glycolysis is a set of reactions that convert glucose into two pyruvate molecules, while gluconeogenesis is the production of glucose from non-carbohydrate sources. The raw material is glucose in glycolysis, while in gluconeogenesis the raw material is lactate. Amino acids like alanine and glycerol occurs in the cytoplasm of all the cells, while gluconeogenesis occurs in the mitochondria and cytoplasm. Glycolysis is a catabolic process, while gluconeogenesis is an anabolic process. Glycolysis is exergonic reaction, while gluconeogenesis is endergonic reaction. Glycolysis occurs through 10 reactions, while gluconeogenesis occurs through 2 essentially irreversible reactions in the glycolytic pathway are bypassed by 4 new reactions. Now, here are some clinical relevances of gluconeogenesis. Diabetes. Overactive gluconeogenesis converts into high blood sugar level. Alcohol excess will cause blocked gluconeogenesis and hypoglycemia. Balance between pathways. It is important to keep the balance between both the pathways. Glycolysis breaks glucose, it means energy is used. Gluconeogenesis makes glucose, it means energy is supplied. Controlled by the hormones like insulin. Glycolysis is controlled by the insulin, and glucagon and cortisol activates the gluconeogenesis. Here is the brief summary of the whole discussion. Glucose, it's a fuel which runs the life. Glycolysis is basically the breakdown of glucose to form the ATP, which is the energy currency. Gluconeogenesis is the breakup system which forms the new glucose. Together, they maintain the blood sugar level. So now we are able to know that glucose is the life's energy currency. Like, share and subscribe to my channel Thinker Zone, and comment below in the comment section should I cover the Krebs cycle next. Thank you for watching the video.